Welcome to Ultimate Sleep Doctor. Tonight, we'll journey to a place hidden deep within the Earth. Beneath our feet, in the dark, silent spaces between rocks, lies a world we've only just begun to understand. It's a biosphere teeming with microscopic life, a secret ecosystem sustained by chemical energy rather than sunlight. These resilient organisms, bacteria and archaea, have carved out an existence in a realm of constant pressure and darkness. Perhaps it's a reminder that life finds a way, even in the most unexpected corners of our planet. A quiet testament to the enduring power of adaptation and the profound interconnectedness of all things. If you find these explorations soothing, please consider liking this video and subscribing to Ultimate Sleep Doctor. Together, we can soothe more wandering minds, one peaceful night at a time. Just breathe. Let your thoughts drift like clouds across a twilight sky. Let's begin. Deep beneath the ocean's surface, far beyond the sunlight, lies a world unlike any we've ever known. It's a realm of perpetual twilight, of crushing pressure, and astonishingly of life. But this life isn't fueled by sunshine. It's built upon a very different foundation, one forged by the Earth itself. To understand how life could thrive in this hidden world, we need to explore the geological processes that created it, starting with the incredible environments found along the Mid-Atlantic Ridge. This ridge isn't a gentle, rolling landscape. It's a massive, underwater mountain range, constantly being built as the Atlantic Ocean's tectonic plates slowly pull apart. This separation isn't just a geological event, it's the engine driving the creation of hydrothermal vent systems, some of the most extreme environments on our planet. These vents aren't volcanoes in the traditional sense. They're cracks in the Earth's crust, fueled by the molten rock deep below. As the plates drift apart, this molten rock rises, interacting with the surrounding seawater. The rocks involved in this process are particularly fascinating. Much of the ridge is composed of mafic rocks, rocks rich in magnesium and iron, formed from the cooling of magma. But perhaps even more crucial are the serpentinites, rocks formed from serpentine, a mineral created when olivine, another common volcanic rock, reacts with water. Serpentinite's unique structure is like a sponge, incredibly porous and capable of holding vast amounts of water. The interaction between these rocks and seawater is where the magic and the potential for life begins. Seawater, brimming with dissolved minerals and gases, seeps into the cracks in the rock. The heat from the rising magma, combined with the pressure, forces this water down into the rock, creating a complex and dynamic fluid flow system. This isn't a simple flow, it's a chemical reaction zone. The water reacts with the serpentinite, dissolving its minerals and, importantly, releasing hydrogen. Hydrogen, a tiny molecule, is a potent energy source. It's a key component in the metabolic pathways of many microbes, allowing them to thrive without sunlight. The reactions also generate methane, ammonia, and other compounds. These chemicals, along with the dissolved hydrogen, form the basis of a chemosynthetic food web, a web where energy comes from chemical reactions, not sunlight. The serpentinite also contains trace metals, like iron and manganese, which are incorporated into the metabolic machinery of these microbes. The process doesn't happen overnight. We're talking about timescales of millions, even billions of years. The Mid-Atlantic Ridge and similar vent systems around the world have been operating for tens of millions of years, providing a stable, chemically rich environment for microbial communities to flourish. These vents existed long after the last traces of surface life disappeared, suggesting that life may have originated and evolved in these deep, dark places. The continuous circulation of fluids, the constant renewal of chemical energy, and the supportive mineralogy of the rocks created a sanctuary, a hidden biosphere sustained by the very heart of our planet. It's a remarkable story of resilience, adaptation, and the enduring power of life. The quest to understand life beneath our feet began not with looking, but with listening. Scientists realized that traditional methods, growing microbes in the lab, were failing spectacularly when it came to the deep subsurface. The conditions down there, immense pressure, frigid temperatures, and a scarcity of readily available nutrients, made cultivating these organisms incredibly difficult, often resulting in them dying before they could even show their faces. So, a new approach emerged: metagenomics. It's like sending a tiny, molecular detective down into the darkness to survey the microbial inhabitants without needing to capture and nurture them. The first real breakthroughs came from samples extracted from incredibly deep boreholes, like the Kola superdeep borehole in Russia. Scientists drilled down over 2 miles, and what they found was astonishing: a vibrant, albeit hidden, world. The key technique involved sequencing the genetic material directly from the rock samples. Specifically, they used 16S rRNA gene sequencing, a universal marker found in nearly all bacteria and archaea. This gene acts like a barcode, allowing researchers to identify the different microbial groups present, much like classifying different species of animals. The initial data revealed a surprisingly high diversity, far exceeding what was expected. Alongside familiar bacteria, a vast number of archaea, many previously unknown, were detected. But 16S rRNA gene sequencing only provides a glimpse. To truly understand the metabolic capabilities of these communities, scientists then employed shotgun metagenomic analysis. This involved sequencing the entire DNA content of the sample, generating a massive dataset of genetic information. From this, they could identify not just the types of microbes present, but also the genes involved in their metabolism, the chemical processes they use to survive and thrive. The data painted a picture of organisms exquisitely adapted to their extreme environment. Particular attention was paid to metabolic pathways. Sulfur metabolism was incredibly prevalent, reflecting the abundance of sulfide minerals found in these deep rock formations. Bacteria and archaea were identified with specialized enzymes designed to utilize sulfate as an energy source. Similarly, iron metabolism was widespread, with organisms possessing the machinery to oxidize ferrous iron, a common component of the rocks. These weren't just simple microbes. They had evolved unique enzymes capable of functioning under immense pressure and at near-freezing temperatures. The relative proportions of different microbial groups also offered insights. Some communities appeared dominated by archaea, while others favored bacteria. Analyses of the DNA sequences revealed that certain groups were particularly adept at scavenging scarce resources, perhaps utilizing carbon from decaying organic matter that had slowly seeped down from the surface over geological timescales. These communities weren't isolated. They were linked through complex biogeochemical cycles, interacting in ways scientists are still unraveling. The Kola superdeep borehole, alongside samples collected across continental shields, became a treasure trove of information. Each new analysis revealed new genes, new enzymes, and a deeper appreciation for the tenacity and adaptability of life. It showed that even in the deepest, darkest corners of our planet, life wasn't just surviving, it was thriving, quietly shaping the very geology beneath our feet. The more we learn, the more we realize how interconnected everything truly is. Deep beneath our feet, a hidden world thrives, fueled not by sunlight, but by the very rocks themselves. This is the realm of chemosynthesis, a remarkable process where microbes become tiny, tireless engines, converting chemical energy into life. Instead of chasing photons, these organisms, primarily archaea and bacteria, specialize in oxidizing reduced inorganic compounds, essentially eating rocks to survive. Consider the immense potential energy locked within molecules like hydrogen sulfide, H2S, methane, CH4, and ferrous iron, Fe2. The process begins with the oxidation of these compounds. For instance, bacteria can use hydrogen sulfide, produced through the interaction of seawater with serpentinite rocks, as an electron acceptor during respiration. This reaction releases energy, which the microbes then harness to fix carbon dioxide, much like plants, but without needing sunlight. The overall reaction, simplified, looks like this: H2S plus 2O2 right arrow, so 2 plus 2H plus energy. Methane, often generated from the breakdown of organic matter in the subsurface or produced by specific archaea, serves as another vital energy source. Similarly, iron, abundant in the form of Fe2 within iron-rich rocks, can be oxidized by bacteria, releasing electrons and creating a cascade of reactions. The reactions are complex, involving numerous intermediate steps, but the fundamental principle remains. The microbes are taking electrons from these reduced compounds and using that energy to fuel their metabolism. A particularly important role is played by iron oxides, like hematite, Fe2O3, which are incredibly common in subsurface rocks. Bacteria can oxidize Fe2 to Fe3, a process often catalyzed by enzymes. This isn't just a simple chemical reaction, it's a remarkably efficient electron acceptor, contributing significantly to primary productivity. Geochemical models, utilizing reaction rates, typically expressed as reaction orders, and concentrations of reactants, allow scientists to estimate the potential energy available. For example, a higher concentration of Fe2 will generally lead to a faster reaction rate and thus greater energy production. The mathematical framework isn't just theoretical. Researchers can calculate the theoretical maximum production rate of organic carbon based on the availability of reactants and the efficiency of the enzymes involved. Furthermore, the pH of the environment dramatically influences reaction rates. Acidic conditions generally favor these oxidation reactions. Estimates vary wildly depending on the specific environment. A hot, acidic vent system will likely support vastly different microbial communities and production rates than a cold, neutral environment. Modeling considers factors such as temperature, pressure, and the presence of other minerals, all of which can impact these chemical reactions. These calculations aren't perfect, of course, but they provide valuable insight into the energetic potential of the deep biosphere. Ultimately, the chemosynthetic microbes of the deep biosphere represent a fundamental pillar of life, demonstrating that energy can be derived from sources other than the sun, and that life can flourish in the most unexpected corners of our planet. Their continued activity shapes the very geochemistry of the deep subsurface, ensuring the continuous cycling of elements and potentially contributing significantly to global biogeochemical cycles. Deep within the Earth's crust, a hidden world thrives, one sculpted not by sunlight, but by the tireless activity of microbes. These aren't simply organisms clinging to life. They've evolved a remarkable partnership with the very rocks around them. The key to this collaboration lies in the mineral surfaces, specifically the way microbes interact with these materials, utilizing them as catalysts and even as a source of sustenance. Consider pyrite or iron sulfide, Fe(S)2, a common mineral formed when iron and sulfur combine. Its surface isn't just rock, it's a tiny, incredibly active platform for microbial metabolism. Certain bacteria, particularly those that thrive in environments rich in pyrite, possess enzymes specifically adapted to oxidize iron. This isn't a passive reaction. The mineral surface itself dramatically accelerates the process. The enzyme, typically iron oxidation reductase, binds directly to the pyrite crystal, facilitating a much faster and more efficient transfer of electrons. The reaction produces ferrous iron, Fe2, and hydrogen gas, H2, both valuable compounds for the microbes. But it's not just pyrite. Clay minerals, like montmorillonite, also play a vital role. These layered structures provide surfaces for microbial attachment and, crucially, for the storage and transport of ions. Bacteria can utilize the exchange sites within the clay to concentrate elements like magnesium or calcium, effectively increasing their local availability. This isn't about simply existing on the clay. It's about participating in a localized geochemical cycle. Furthermore, microbes frequently form biofilms, complex communities of bacteria encased in a self-produced matrix, directly on mineral surfaces. These biofilms aren't random collections, they're highly organized, with different bacterial species fulfilling specialized roles. Some might be focused on oxidizing sulfur compounds, others on precipitating iron oxides, and still others on utilizing the byproducts of these reactions. The mineral surface acts as a scaffold, providing the structural support and promoting the exchange of nutrients and waste products within the community. The composition of the mineral surface itself profoundly influences the microbial community. Different minerals have different surface chemistries and crystal structures, which directly affect the types of enzymes that can bind and the metabolic pathways that can be sustained. For instance, a sulfide-rich environment will support sulfur-metabolizing bacteria, while an iron-rich environment will favor iron-oxidizing organisms. The interplay between mineralogy and microbial activity creates a dynamic mosaic of metabolic niches. This doesn't just affect the diversity of individual species, it shapes entire microbial communities. The ability of a microbe to thrive in a particular subsurface environment is fundamentally linked to its capacity to interact with the surrounding rock. The remarkable tenacity of life within the deep biosphere hinges on this intimate connection, demonstrating that even in the absence of sunlight, life can find a way to flourish, powered by the chemistry of the Earth itself. The deeper we go beneath the Earth's surface, the stranger things become. It's not just cold and dark, though those are certainly factors. The real challenge for life in these hidden realms is the pressure, an incredible weight pressing down from every direction. Imagine a world where every drop of water, every molecule of gas, is squeezed with immense force. This pressure isn't a passive force. It actively alters the very building blocks of life. Cellular membranes, normally flexible and fluid, face a serious struggle. At great depths, the lipids that make them up aren't as easily arranged. Scientists have identified microorganisms, we call them piezophiles, that have evolved specific adaptations. They produce lipids with longer chains, creating membranes that are much more rigid and resistant to compression. Think of it like building a wall rather than a bouncy castle. The membranes are built to withstand the relentless push. Protein stability is another critical hurdle. The increased pressure increases the forces acting on proteins, potentially disrupting their delicate three-dimensional shapes. Most proteins would unravel under this strain, rendering them useless. However, some microbes possess remarkably stable proteins. They do this by strengthening the bonds within their structure, often by adding more proline, an amino acid known for its rigidity. These protein adaptations are essential for enzymes to function properly, driving the chemical reactions that sustain life. DNA replication also presents a significant problem. The pressure makes it harder for DNA polymerase, the enzyme responsible for copying DNA, to bind to the template strands. Piezophilic microbes have evolved specialized DNA polymerases with altered mechanisms to overcome this challenge, often incorporating structural adaptations within the enzyme itself. These adaptations ensure accurate DNA replication, vital for inheritance and survival. The research into these adaptations isn't just confined to subsurface environments. Studying piezophiles in the deep subsurface offers a valuable comparison to life around hydrothermal vents at the bottom of the ocean. Both environments represent extremes of pressure, though the temperature differences are significant. While vent microbes are adapted to intense heat and chemical gradients, subsurface microbes demonstrate an equally impressive ability to thrive under crushing pressure. Interestingly, the mechanisms employed by subsurface microbes often mirror those seen in vent ecosystems, suggesting a shared evolutionary history and common strategies for survival in similarly harsh conditions. The chemical gradients, like the oxidation of methane or iron, remain crucial energy sources regardless of the location. Scientists are continuing to uncover novel enzymes with remarkable pressure resistance, offering potential applications in biotechnology and materials science. These extreme enzymes could revolutionize industries requiring stability under high pressure, from deep-sea exploration to the development of new pharmaceuticals. It's a reminder that life, in its tenacity and adaptability, continues to surprise us, even in the most unlikely corners of our planet. The deep subsurface is a surprisingly busy place, a hidden world where life persists not through sunshine, but through a constant exchange of materials. It's a system built on movement, the flow of water, gases, and, most critically, the transport of nutrients. Think of it like a vast, slow-moving river system beneath our feet, only instead of water, it's primarily dissolved gases and tiny particles of organic matter. The key to this movement are the fractures and porous rocks that crisscross the Earth's crust. These pathways act like conduits, delivering fresh supplies of nutrients, like hydrogen, methane, and phosphate, to microbial communities and whisking away the byproducts of their metabolic activities. The movement isn't random. It's driven by pressure gradients, the difference in concentration of nutrients between areas of high and low abundance. Water, drawn by these differences, slowly percolates through the rock formations, carrying dissolved gases and nutrients with it. Importantly, the shape and interconnectedness of these flow pathways, from microscopic pores to kilometer-long fracture systems, dramatically affect the distribution of these resources. Some pathways are incredibly narrow, creating localized zones of high nutrient concentration, while others form extensive networks, facilitating wider dispersal. Microbes themselves play a significant role in nutrient transport. They often form biofilms, sticky, layered communities of microbes attached to mineral surfaces or even to each other, which act like miniature conveyor belts. These biofilms can dramatically increase the surface area available for nutrient uptake and release. Furthermore, many microbes secrete exopolysaccharides, complex sugar-like molecules that create a gel-like matrix around themselves and their neighbors. This matrix facilitates the movement of nutrients throughout the biofilm and allows for the efficient capture of dissolved materials from the surrounding water. Consider the transport of phosphate. It's often a limiting nutrient in these deep environments, and microbes have evolved clever strategies to acquire it. They might actively pump phosphate ions across their cell membranes, or they could bind the phosphate to iron oxides, a process that effectively traps the nutrient within the microbial community. Similarly, the movement of iron is tightly linked to microbial metabolism. Iron oxides, like those found in pyrite fool's gold, are frequently used as electron acceptors by bacteria, and the microbial communities around these minerals become hotspots for iron cycling. Scientists are increasingly using computer models and simulations to understand these complex nutrient cycling processes. These models take into account diffusion, the movement of nutrients due to concentration differences, advection, the movement of water carrying nutrients, and, crucially, the metabolic activity of the microbes themselves. By simulating these processes, researchers can predict how nutrient distributions will change over time, how microbial communities will respond to environmental fluctuations, and even how these processes might impact the global carbon cycle. These sophisticated models demonstrate that the deep biosphere isn't just a passive recipient of nutrients, but an active participant in shaping the geochemistry of the Earth. The Earth's crust isn't just rock and dirt. It's a hidden world, a vast, interconnected network of tunnels and chambers stretching miles beneath our feet. And within this darkness, a remarkable story unfolds, one that suggests life might be far older and far more resilient than we ever imagined. The deep biosphere, sustained by the energy and nutrients locked within these rocks, could represent a living fossil record, a testament to life's tenacity. Recent discoveries are fueling the idea that life didn't simply appear on the surface, but instead established itself deep below, shielded from the dramatic shifts and catastrophic events that have repeatedly challenged surface life. Scientists have found evidence of surprisingly complex microbial communities thriving in extremely ancient rock formations, rocks that are billions of years old. The presence of active metabolism in these samples is astonishing. It suggests that life may have been quietly persisting, using the chemical energy present in minerals to fuel its existence, long before the conditions on the surface were suitable for complex life forms. Furthermore, the discovery of intact DNA fragments within these ancient rocks is incredibly significant. While degraded, The presence of genetic material indicates that the microbial communities weren't just dormant. They were actively replicating and evolving, albeit slowly, over immense spans of time. This raises a tantalizing question. Could the deep biosphere be a backup for life on Earth? A refuge where life could have survived periods of intense bombardment, volcanic activity, and climate change that wiped out surface life. It's a compelling scenario, one that aligns with the idea of planetary resilience. And it's not just about Earth. The concept of panspermia, the possibility that life originated elsewhere in the universe and was then transported to Earth, gains even more weight with these discoveries. Asteroids and comets, those rocky travelers of the solar system, are rich in organic molecules, the building blocks of life. Impacts from these celestial objects could have delivered these molecules to Earth, perhaps even carrying microbial life itself. While definitively proving panspermia is incredibly challenging, the evidence is becoming increasingly suggestive. Scientists are analyzing the isotopic signatures of microbes found in deep subsurface environments, comparing them to those found in meteorites. The alignment of these signatures strengthens the argument for an extraterrestrial origin. Moreover, the deep biosphere may have played a crucial role in the very early evolution of life on Earth. If microbial communities were thriving in the deep subsurface from the planet's infancy, they could have been responsible for key biochemical transformations, such as the formation of oxygen, that ultimately paved the way for the emergence of more complex life forms on the surface. The deep biosphere, therefore, isn't just a hidden ecosystem. It could be a foundational element in the story of life itself, a story that extends far beyond the familiar world above. As we continue to explore these hidden realms, we're not just discovering new microbes, we're unraveling the deepest secrets of our planet's history, and perhaps our place within the cosmos. The quest to understand the deep biosphere is rapidly evolving, fueled by increasingly sophisticated technologies designed to peek into these hidden realms. Currently, the most direct approach involves pushing the boundaries of drilling, and it's proving to be a remarkably challenging endeavor. Traditional borehole drilling simply isn't adequate. The pressures and temperatures plummeting with depth create immense difficulties for equipment. We're seeing the development of advanced deep boreholes, often utilizing remotely operated vehicles, ROVs, to guide and monitor the drill bit as it descends through kilometers of rock. These boreholes aren't just about creating a hole, they're about creating a controlled environment to study what's found within. Alongside borehole development, submersible drilling systems are gaining traction. These systems utilize specialized pumps and cutters to slowly erode rock formations underwater, offering a potential pathway to access hydrothermal vents and associated subsurface environments directly. The ability to manipulate the drilling process remotely is critical, allowing scientists to adjust for unexpected geological conditions and maintain the integrity of the borehole. Another exciting development is the exploration of swimming drills vehicles designed to navigate the subsurface by essentially swimming through the rock, perhaps utilizing microfluidic systems to push themselves forward. However, simply reaching the deep subsurface isn't enough. We need to characterize the environments we've accessed. Geochemical logging, using instruments lowered into boreholes, allows us to measure temperature, pressure, and the concentration of various chemical compounds, everything from dissolved gases to trace metals. These measurements paint a picture of the conditions within and help scientists understand the potential for microbial activity. Sophisticated geophysical surveys, utilizing seismic waves and gravity measurements, are also playing a crucial role, mapping subsurface structures and identifying potential fluid pathways. Looking ahead, miniaturized sensors are poised to revolutionize real-time monitoring. These tiny devices, equipped with electrodes, temperature probes, and chemical sensors, can be deployed within boreholes to provide continuous data streams. Imagine hundreds of these sensors networked together, relaying information about the metabolic activity of microbes or the flow of fluids in real time. Robotic probes and autonomous systems are becoming increasingly important for exploration and sample collection. Small, remotely controlled vehicles equipped with robotic arms can navigate complex subsurface environments, collecting soil and rock samples for analysis. Furthermore, autonomous systems, perhaps resembling small submarines, can be programmed to systematically explore specific areas, collecting data and samples without direct human intervention. The challenge lies in designing these systems to withstand the extreme conditions and to operate effectively in environments with limited visibility and communication. Developing robust communication protocols for these deep subsurface probes is a significant hurdle. Ultimately, the future of deep biosphere research hinges on a combination of these approaches: drilling, remote sensing, and autonomous exploration. It's a long, iterative process, reaching down, gathering data, adapting our techniques, and pushing the boundaries of what's possible. The potential rewards uncovering entirely new ecosystems and perhaps even unlocking secrets about the origins of life are truly remarkable. If you're still awake and enjoyed this exploration, please consider liking this video and subscribing to Ultimate Sleep Doctor. Together, we can soothe more wandering minds, one peaceful night at a time. We've journeyed deep within the earth, discovering life's tenacious hold within the rock. Allow your thoughts to drift, calm and unhurried, like silent stones settling into the darkness. Let sleep find you, gently and sweetly. Good night.